Welcome to this week's episode of Ask an MS Expert. I'm John Strum, and I'm the host of the Real Talk MS podcast. Each week, Real Talk MS reaches thousands of people in more than 100 countries around the world with the news that people affected by MS need to know. My wife, Jean, lived with progressive MS for 23 years, so I've had a front row seat experiencing all the ways that MS can impact a family. Currently, I'm a member of the International Progressive MS Alliance Scientific Steering Committee. I'm a district activist leader and trustee for the National MS Society, and I chair the Society's California Government Relations Advisory Committee. As we get underway, I'd like to acknowledge and thank today's national premier sponsor, EMD Serono, and our national sponsors, Humana Specialty Pharmacy and Janssen. Support from MS community partners helps the National MS Society reach its goal of providing up-to-date information and support through programs like Ask an MS Expert. Thank you to all of you who are joining us on GoToWebinar, Facebook Live, and YouTube Live. The MS Society's Ask an MS Expert webcast is designed to connect you with MS experts who are ready to answer your questions on the topics that impact people affected by MS every day. So as I chat with our expert today, please feel free to post your questions on Facebook and YouTube, or type them into the question box if you're joining us on GoToWebinar. We'll try to answer as many of your questions as we can during the Q&A portion of today's program. We're at a moment in science where science is making significant progress, and breakthrough solutions hold the promise of changing the world for everyone living with MS. Today, Dr. Michael Kornberg is joining us for a discussion about innovative approaches to treating MS. Dr. Kornberg is an assistant professor of neurology and associate director of the neurology residency program at the Johns Hopkins University School of Medicine. He's a member of the Johns Hopkins Immunology and Cellular and Molecular Medicine graduate programs, and Dr. Kornberg specializes in the care of individuals with multiple sclerosis and other autoimmune diseases of the nervous system. In addition to his clinical work, Dr. Kornberg runs a research program focused on better understanding the pathogenesis of MS and identifying novel therapeutic strategies to prevent neurodegeneration and promote remyelination in MS. Welcome, Dr. Kornberg, and thank you for being with us today.
Thank you very much for having me, John. It is really my pleasure, and thank you to everyone who's tuning in.
New attempts to target MS progression are focusing on B cells and innate immune cells, also called myeloid cells. Dr. Kornberg, can you explain to our viewers the role of these cells in MS progression?
Absolutely. So I'll talk about them separately first. So B cells are a type of immune cell in our body that are best known for their role in becoming antibody-producing cells. But it turns out that they seem to do a lot of other important things in our immune system, including interacting with other types of immune cells like T cells to help them take on some of their inflammatory activities. And we know now that B cells do play a role in relapses and in relapsing remitting MS. And that work is what led to some current therapies like ocrelizumab that target B cells and deplete them. And we know that those are very effective therapies at treating relapsing remitting MS. There is some reason to believe that B cells might also play some roles that are relevant to progressive MS. For instance, in people with progressive MS, we see inflammation, not just within the brain, but we start to see inflammation in the covering around the brain, something that's called the meninges. And some of that inflammation consists of these collections of B cells. And underneath those collections of B cells, we see damage to the outer part of the brain and demyelination and things that we think are relevant to progressive MS. And so there's some thought that B cells, through their role in this what's called meningeal inflammation, may play a role in progression. On the other hand, what we call innate immune cells, they also play roles both inside the brain and outside the brain. So outside the brain, they're responsible for activating cells like B cells and like T cells. And so they also are relevant for relapsing remitting MS. But as some of the listeners may know, in progressive MS in particular, there's less inflammation happening outside the brain, and it kind of gets trapped inside the brain. And these certain types of cells called microglia and macrophages seem to play a particular role in the progressive phase of the disease. And so these cells have traditionally not been targeted by the drugs that are currently on the market and the other therapies we have for MS. And so they're a type of cell that's of great interest in terms of halting or at least slowing progression.
There's a new class of drugs being tested to treat MS called BTK inhibitors. These drugs inhibit a specific enzyme which reduces B cells and myeloid cells activation. Dr. Kornberg, can you explain how these BTK inhibitors work?
Absolutely. So just like you said, the Bruton tyrosine kinase is a protein that B cells and also some of these innate immune cells have on their surface. And these drugs, just like the name of them, they're called BTK inhibitors. They inhibit this particular protein, which prevents B cells and innate immune cells from becoming activated. And so because of that, they certainly have relevance in relapsing remitting MS. And as opposed to other therapies like ocrelizumab that deplete B cells, these BTK inhibitors have kind of a fast action of on and off, and so they work very quickly. But when you stop taking them, the B cells return to their normal function very quickly. But unlike other medications that target B cells, they also target these innate immune cells that play roles both in relapsing MS and in progressive MS.
Some BTK inhibitors have been shown to penetrate the blood-brain barrier. What makes that important in treating MS?
Yeah, so what makes that important, in my view, is the sort of promise that a therapy might hold for progressive forms of MS. So in relapsing MS, we know that all of the action initially happens outside the brain. The immune system gets activated in lymph nodes, in the blood, and it crosses into the brain to create the damage. In progressive MS, all of that, the inflammation that persists, all of that seems to be kind of trapped within the brain. And so in order to have an impact on that, we have to have drugs that cross into the brain. So there's something called the blood-brain barrier, which is part of our body that's designed to keep bad things outside of our brain. But it also is a big challenge when it comes to developing therapies, medications that can have an impact on progressive MS, where they need to get into the brain. And so because these drugs have an effect on the innate immune system, which we think is important for progressive disease, if they can penetrate the brain, then they have a chance of impacting the disease in that way. And there are currently trials either underway or recruiting for a variety of these BTK inhibitors. And the ones that are brain penetrant, there are some studies in the works looking specifically at progressive MS.
Now, one of the BTK inhibitors, called Evobrutinib, is currently being tested for treating MS. What do preliminary results show about this particular drug?
Yep, so this drug is probably the farthest along in development of the BTK inhibitors, and it was looked at in what we call a phase 2 study of people with relapsing remitting MS. And phase 2 study is kind of, it's just before prime time is the way I like to think of it. The big study that ends up leading to a drug getting approved is called a phase 3 study. And so phase 2 is one step before, but still involves a sizable number of people. And what was found was that Evobrutinib, when given to people with relapsing remitting MS, does decrease new lesions forming on MRI and does decrease relapses. And so that is known, and it's moving on to phase 3 therapies, and I think it might certainly have a role in relapsing remitting MS. What remains to be seen, both for Evobrutinib and other drugs in that class, is whether it might have an effect in progressive MS.
Well, while we've been talking about these particular drugs, I've heard you talk about both relapsing and progressive forms of MS. So is this a type of therapy that'll be beneficial or potentially beneficial to both relapsing and progressive forms of MS?
So potentially, yes. I am a cautious optimist, I guess, by nature. And I would say I have little doubt that this class of therapy will be effective and have a role in relapsing MS. Progressive MS is going to be a higher bar, and so it's certainly I feel less certain about what their ultimate role will be. But just based on what we know about the cells that they impact, particularly the drugs that get into the brain, I am excited about a potential role in progressive MS, and we have to wait for the trials to see. But I could certainly see this class of drugs having a role there.
Researchers are also making progress in understanding how the body's gut microbiome may impact MS. Can you tell us what the gut microbiome is and what we're learning about its role in multiple sclerosis?
Yeah, so this is an area of research that has really exploded over the last really 5 years. It's very exciting. In some ways, it's still in early stages. So what the gut microbiome is, we all have trillions of bacteria that are colonizing our gastrointestinal tract, our intestines. I think the longest period of medicine, they were thought to just be just sitting there helping us digest food, but not really playing much of a role. And so what we've found is that the composition of the microbiome, meaning the specific types of bacteria that are there, have a relation to various aspects of multiple sclerosis and other conditions. And they seem to do that in a number of ways. So they have relevance to the function of the immune system and to inflammation specifically. And so the composition of the types of bacteria that are in our gut plays a role in determining the immune system that is present within the gut itself. And we know that immune system is relevant even in multiple sclerosis. So if you have more pro-inflammatory, anti-inflammatory cells forming in the intestines, those end up contributing to the inflammatory environment in the rest of the body and in the brain. At the same time, all of those bacteria, they produce different chemicals themselves, from the food that we eat. And some of those chemicals that are unique to different types of bacteria seem to have an effect on cells even within the brain. And so this has been, this work has been more in kind of the animal study stages than in humans. But there's been some outstanding work by people like Francisco Quintana at Harvard that's shown that some of these chemicals that are produced by specific bacteria can get into the brain and change the way some of the important inflammatory cells in our brain behave.
I think Dr. Quintana is showing us that the way to the immune system may in fact be through the gut.
Right, like all good things in life, right?
Exactly. We've heard from Caroline, who's asking, what has research found about how gut bacteria may impact MS progression?
Yeah. So what we were just discussing with Dr. Quintana, I think is the most relevant. I think with, like everything, what we know in terms of relapsing, remitting MS is a little bit further along than with progressive MS. But we do know that the composition of the gut microbiome and the specific chemicals that are produced by them impact processes that we think are specifically important for progressive MS. And so when we talked about the innate immune system, I mentioned a few cell types, things that are called microglia and macrophages. But there's another cell in the brain, something called an astrocyte, that we now know seems to play a big role in kind of the inflammatory environment that's present in the brain, seems to have functions that we think are very relevant specifically to progressive MS. And some of these chemicals produced by bacteria, specifically chemicals that are breakdown products of a type of a component of protein, something called tryptophan, they have specific effects on these astrocyte cells, which we think are relevant to progressive MS. You know, there's a big chasm between, you know, the way mice work and the way people work, and you know, obviously we have to be cautious about making these extrapolations. So it remains to be seen, you know, what the relevance is specifically in progressive MS. But there is a lot of promising and suggestive research at the preclinical level, meaning before it's gotten to people.
Being able to accurately predict an individual's disease course and MS progression requires developing reliable biomarkers. Can you tell us what a biomarker is and what its role is in diagnosing, predicting, and managing MS?
Yeah, so a biomarker is something that we can easily, non-invasively measure in a person, that either provides certainty about diagnosis or tells us something about how their disease is behaving. In MS, you know, we don't have a lot of good, you know, what I would call, you know, biomarkers that provide a definitive diagnosis. You know, which is why the diagnosis is put together through, you know, information about, you know, the symptoms someone's had, you know, the pictures from their MRI, you know, maybe spinal fluid. But biomarkers are now, I think, very much on the horizon in terms of telling us how active someone's MS is, what their risk is of future disability, and also biomarkers that will help us to design clinical studies to find agents that work in things like progressive MS. So, you know, as you can imagine, to look at disability in a study of progressive MS can take years. But a biomarker that gives you more information about what sort of damage is happening to the brain can allow you to know whether your treatments are working in a much shorter time frame.
Neurofilament light chain is being studied as a predictive biomarker of MS disease activity and disability progression. Can you tell us what this is and how it could be used in diagnosing MS or predicting MS progression?
Yeah, absolutely. So some of the listeners joining, you know, might be aware of this focus on brain atrophy in multiple sclerosis. And really that's a measure of, you know, whether the nerve cells themselves are healthy or whether they're kind of dying off and degenerating. And, you know, we talk about multiple sclerosis as a demyelinating disease, meaning that, you know, myelin is kind of, you know, what's destroyed, but it turns out that, you know, myelin is coating these nerve cells, that the nerve cells themselves actually experience significant injury, both in relapsing remitting MS, but particularly in the progressive phase of the disease, we start to see injury to nerve cells themselves. Neurofilament light is a protein that is found only in nerve cells. So it's in these long, you know, the long processes called axons that help nerve cells transmit their signals. And so when a nerve cell is damaged, it releases this protein and we can measure it. And so it basically tells us whether there is active damage to nerve cells, because if these nerve cells are healthy, that neurofilament light is staying within them. We can't measure it in their spinal fluid or in their blood. It used to be that you had to get spinal fluid from someone, do a spinal tap to measure neurofilament light. But now we have much more sensitive assays that allow us to measure them from blood. And so it's a biomarker that tells us how those nerve cells are being damaged, which really is ultimately the biggest source of disability for people with MS. There's a few ways in which that biomarker is being used and may be used in the future. So when someone is having a relapse, you have a lot of damage to those nerve cells, and so the levels of neurofilament light go up. And so this is being used as a biomarker to tell us how active the inflammatory part of your MS is. And these days, if you're not having an attack, it's not easy to get an MRI, right? And we get MRIs every 6 months, most frequently, but for many people it's once a year. So how do we know how your disease is doing in those intervals? And this is a simple blood test that gives us some of that information. And so it's going to play a large role in making sure that the treatment that we have people on is adequately controlling their disease. We've also seen that the people who have higher levels of neurofilament light in their blood are at greater risk for future disability, which is not surprising, right? Because it means that they have more active damage to their nerve cells. And so in helping us decide what therapy is right for what person, it gives us some information about how they're likely to do in the future. And then finally, it might be a very useful tool when we are designing clinical studies for people with progressive MS, because the hope is that when you can impact that injury to nerve cells, which really is kind of the crux of the problem in progressive MS, that it gives us one of those biomarkers we can study in clinical trials, so we don't have to wait 2 years to measure something like disability.
Some novel approaches in MS research are based on targeting viruses that might be linked to MS. Previous infection with the Epstein-Barr virus has been linked to the risk of developing MS. What are we learning from recent studies focused on the Epstein-Barr virus and MS?
Yep. So Epstein-Barr virus infection is one of the best known and best studied risk factors for developing MS. And the data that we have, not as recent, but not that long ago, tells us that being infected with Epstein-Barr virus is almost a prerequisite for developing MS. And there have been very well done studies in groups of people like kids, where so kids can develop MS. And there have been studies in which groups of kids had blood samples from their pediatrician before, and when they developed MS, they could go back and look at those blood samples, and they found that kids only developed MS after they've shown signs of being infected with Epstein-Barr virus, meaning they had antibodies in their blood. There have been similar studies done in military recruits where they had blood samples over time, and they found that people don't develop MS until after they've been exposed to EBV. I think more recently, the most interesting data has been that there was this idea that some kids, maybe 15% of kids who develop MS, did not have evidence of prior Epstein-Barr virus infection. And that was kind of always a little bit of contradictory evidence, but there was a very recent study from Barty Norbosch, who's at Johns Hopkins, and Emmanuel Wobont, who's at UCSF, where they looked at those kids that did not show signs of prior Epstein-Barr virus infection. And when they really look at their disease, they find that most of those kids didn't have MS. They had related conditions. And so it does seem like if not 100%, nearly 100% of people who have MS have been exposed to the Epstein-Barr virus. The tricky thing is that if you look at a group of people who do not have MS, by the time they reach their 20s, about 90% of those individuals show signs of prior exposure to Epstein-Barr virus. So it's kind of it's necessary but not sufficient for the development of MS. And we still have a lot to learn about why exposure to Epstein-Barr virus seems to be related to MS. And there's many possibilities, and some of those possibilities really impact whether treatments targeting Epstein-Barr virus or the body's response to it actually hold promise as a treatment.
A monoclonal antibody called Emelomab is currently being studied. This is an agent that targets other types of viruses called human endogenous retroviruses. What can you share with us about this research?
Yeah, so this is, I think, one of the more fascinating aspects of biology that's relevant to MS. So endogenous retrovirus there are some viruses that the way that they work when they infect a cell, they kind of integrate their DNA into our DNA. But there are some viruses that seem to, sometime in the ancient past have infected, early humans and incorporated their DNA into our DNA in a way that actually just became part of our DNA over time. And so, we have these remnants of these ancient viruses kind of in our cells, which is strange to think about. But these, the DNA, the genetic information from these old viruses produces proteins that we have in our cells. And in particular, there's one protein that has been found in MS lesions, in both relapsing MS and progressive MS. And in some animal studies, targeting this protein has shown benefit. And so this drug, this antibody is specifically targeting this protein that's related to an ancient virus that our bodies have carried forward. The studies being done in humans are kind of very early stages, but it's being looked at both in the context of relapsing, remitting, and progressive MS.
Studies have shown that early and ongoing treatment with a disease-modifying therapy has long-term benefits for controlling disease activity, delaying progression, and improving quality of life for people living with MS. Now, recent clinical trials are evaluating if early and aggressive treatment of MS with DMTs may be more effective in the long term than gradually escalating to stronger treatments over time. What can you tell us about these two different approaches to prescribing disease-modifying therapies?
Yes, so when I talk to patients about available therapies, I kind of put them into three bins. And on one side are the medications that are kind of the gentlest. They're the least effective at stopping attacks, but they generally speaking are also the safest. You have the kind of the least concerns about safety. And then you have the high efficacy therapies, the ones that work the best at preventing relapses, but come with some potential for safety concerns that you have to worry about. And so traditionally, there have been two approaches to treating patients. One is what we call the escalation approach, where you start with one of the safest therapies that we have, knowing that it's not as effective overall, and hope that it's enough for a person. And if that person has breakthrough disease, meaning if they have a relapse or they have new lesion form on MS, then you escalate them to a stronger therapy. On the other end is this idea that, time is brain, and we know that getting control over someone's disease activity early on is important for their long-term disability. So, we should start people on the strongest therapies at the beginning. And as a field, the field has kind of moved more towards the idea of starting people on strong therapies from the beginning, based on, a variety of evidence, of different quality. But the truth is that we just don't know for certain what is the best approach. And when we think about starting people early on strong therapies, we also have to consider the safety aspect as well. And so the trials that you're talking about, there's one called Treat MS, there's one called Deliver MS. They're both taking place at a number of big institutions. Hopkins is participating in Treat MS. And the idea there is to randomize people to one of those two approaches, either the escalation approach or the early high efficacy approach, and see how people do over time, whether there's differences in disability and in safety, so that we really can have a kind of a coherent approach among MS providers to disease modifying therapy.
Before we continue our discussion, I'd just like to take a quick moment to welcome those of you who have continued to join us on GoToWebinar, Facebook Live, and YouTube Live. Please let us know what's on your mind. Post your comments and questions on Facebook and YouTube, or type them into the question box if you joined us on GoToWebinar. MS navigators are also online throughout today's program, answering questions and providing resources. Our Ask an MS Expert live event takes place at this same time every Friday. So please help us make sure that everyone knows about it by sharing news about the webcast with your family and friends and on all of your social media platforms. Dr. Kornberg, there's innovative research focused on the potential of stem cells for slowing MS disease activity and even repairing damage to the nervous system. What are stem cells and what types of stem cells are being explored for their potential benefits in treating MS?
Yeah, so stem cells is obviously an important topic, and it's something that my patients bring up with me quite frequently. So a stem cell, in essence, is a cell in the body that can, on the one hand, renew itself indefinitely, and on the other hand, it has the potential to form a variety of different specialized cells. So it hasn't kind of committed to a specific cell type yet. And in the context of multiple sclerosis, and I know we'll be getting into this topic, I kind of separate them into two categories. So on the one hand, there's the type of stem cell that we hope might actually help to repair the damage that's done to the nervous system, either by directly forming new myelin forming cells, or by promoting that repair process in the brain. And then there are blood stem cells, which is called hematopoietic stem cells, which are not designed to repair damage, but are basically used to replenish the immune system after we kind of get rid of it in a stem cell transplant. So there's the stem cells that we hope are going to participate in repair, and then there's the blood stem cells that play more of a role in kind of rebooting the immune system.
You just mentioned hematopoietic stem cells. There's growing evidence that autologous hematopoietic stem cell transplantation, also referred to more easily as AHSCT, and commonly known as a bone marrow transplant, can be effective for treating some people living with MS. Now, Michael, who's living with relapsing remitting MS, is asking exactly that. What is a bone marrow transplant, and how might that therapy help him?
So the idea with bone marrow transplant is that ultimately the problem with MS, particularly in relapsing remitting MS, is that people with relapsing MS have immune cells floating around in their blood that are self-reactive, that have become reactive against myelin in the brain, and that's really the at the core of what's causing relapses and new lesions on your MRI. And so the idea with bone marrow transplant is essentially like rebooting a computer. The stem cells are collected from your bone marrow and kind of frozen down, and then you're treated with essentially high doses of chemotherapy, which is designed to just ablate your immune system. So all of those self-reactive immune cells you have floating around, it just kind of gets rid of them. And then you're given back your own frozen down stem cells from your bone marrow, which can reform your immune system. And when it's reformed fresh, the hope is that all of those autoreactive T cells and other immune cells are gone, and it's kind of like starting fresh. And so, the whole, the problems in your immune system that are targeting your brain is gone. That's how it works. There are some risks, obviously to the procedure. When you're being treated with high doses of chemotherapy that essentially is intended to ablate your immune system, it means that you are going to be susceptible to infection for a period of time. There's some other issues with some of the regimen that's required. It has some issues with bleeding and with infertility. So there's certainly a downside. But the idea is to kind of reboot the immune system so it no longer reacts against your own brain.
The BEAT MS trial is a large phase 3 clinical trial to study AHSCT. What can you tell us about this study?
Yes, so this study is designed to compare the effectiveness of bone marrow transplant to our other highly effective therapies. because a point that that I think is really important to make, and that I think is not clear to everyone with MS, is that a bone marrow transplant, that type of stem cell transplant, is really targeting the same thing that our current drugs are targeting. They're targeting the immune system outside the brain, and so it works best for relapsing remitting MS. And the question is that the risks of stem cell transplants are tend to be higher than a lot of the medications that we're currently using, and so it's important to know, does it actually work better? And so this study will compare bone marrow transplant to other treatments like ocrelizumab, natalizumab, alemtuzumab, which we consider kind of our most effective current therapies, to tell us whether there really is an advantage to stem cell therapy for people with highly active MS.
The MS Society's National Medical Advisory Committee recently developed recommendations related to the optimal use of AHSCT to treat relapsing remitting MS. According to these recommendations, who are the best candidates for this procedure?
Yeah, so these recommendations, which just like all of the National MS Society recommendations are very thoughtful and well-informed and well-reasoned, are derived from what we've learned from studies of stem cell transplants so far. So I mentioned before that this is really a treatment that's targeting your immune system. And so the people who benefit the most are the people whose immune systems are most active, the people who have the most active forms of relapsing MS, essentially. And so those people tend to be younger, who have had a shorter duration of MS, and who essentially failed at this point, who have failed other strong MS therapies, meaning that they're still having relapses, they're still having new MRI lesions forming despite the best therapy. We know that those are the people who seem to benefit the most from stem cell transplant, and so the National MS Society recommendations are in line with that. What we know from the initial studies that that have been done is that people who have not had a relapse in a long time, are older with progressive MS, at the very least see less benefit, and in many cases maybe not any benefit from stem cell transplant. And so that's kind of at the crux of the National MS Society recommendations. The other part of it that is very important, that's stated in those recommendations, is that stem cell transplant is inherently a risky procedure. There are some centers around the world that have a lot of experience with it, particularly in MS patients. And at those centers, the safety of the procedure has improved dramatically over the last 15 years. But it is extremely important that this is done at a center that has extensive experience with stem cell transplant, bone marrow transplant, as well as MS. And that it's a center that that has been that's regulated and accredited. And so the recommendations from the National MS Society state very wisely that if you're considering bone marrow transplant, you really should be considering it within a controlled clinical study. And so that it refers you to the Beat MS trial, which is looking at this in a very well controlled, very well regulated way. But that if you must go outside of a clinical study, that you need to be very careful that you are going to a center that has lots of experience, and that is accredited by a reputable body. And the National MS Society website links you to those organizations where you can investigate specific institutions.
Other types of stem cells are also being explored for their potential benefits in treating MS. Can you tell us what mesenchymal stem cells are and what the clinical trials are teaching us about the viability of mesenchymal stem cell treatments for MS?
Yeah, so I find mesenchymal stem cells fascinating, and we are at the early stages of clinical studies in looking at the role of these cells. So what these are, they're essentially what I would call multipotent stem cells, meaning they can't form any cell in the body, but they do have the capacity to form a number of different cells of the body. And we get them actually from, from adults. We oftentimes think of stem cells as having to get it from embryos, and these come from adult humans. They're derived from a number of different tissues. You can get them from fat tissue, you can get them from blood. In some animal studies, they've been shown to have a number of roles that can be beneficial for MS. They've been shown to have some anti-inflammatory characteristics, but I think more importantly, in thinking about progressive MS, in some animal models, they've been shown to essentially help other cells of the brain repair damage that's been done. So promote new myelin cells forming, protect nerve cells from dying. And so now we're looking at the role of these cells in humans. The important point to make is that, there's mesenchymal stem cell is kind of an umbrella term. There's not, only one such thing. And so there's many different ways that you can isolate them, that you can treat them before you put them back into the body. It has not been very well standardized at this point. And so, because of that, there are a number of for-profit clinics, that offer mesenchymal stem cell transplant that is not regulated, they're not backed up by evidence at this point, and you don't really have any regulation to tell you exactly what they're taking from you and what they're putting back into you. And so I think it's very important to say that, that there's a wide world of, what's called mesenchymal stem cells. But there are some very well done studies right now being done at multiple institutions around the world, looking at their ability to promote repair and to stop progression. But if you're considering something like that, I strongly urge you to do it within the confines of a clinical study.
Restoring function that's been lost to MS is reliant on, as you just said, myelin repair. So first, can you tell us what causes demyelination or loss of myelin in MS?
So the simplest explanation is that it is a product of the immune attack. So we think that myelin is what the overactive immune system in people with MS is specifically attacking. There may be some other things at play. There's this longstanding idea that in some people with MS, myelin might be just inherently unhealthy, and that triggers the immune response. In progressive MS, there's this idea that other things that can contribute specifically to energy availability, like areas of the brain that aren't getting as much oxygen, can contribute to demyelination. But primarily, it's that this is really the target of immune attack. So when you have a relapse, you lose myelin in the area of the lesion. And later on in progressive MS, where you have this kind of smoldering inflammation, you can have ongoing loss of myelin, both at the edges of old lesions, but also in the outer part of the brain called the cortex, and a little bit more diffusely within the brain.
Now, research shows that the brain is capable of repairing myelin, but in MS, the brain's ability to repair myelin seems to stall. Do we know why this happens, and what are some of the strategies being studied to promote remyelination?
So we don't know definitively why this happens, particularly in any individual patient. But I would say over the past 10 years, we are starting to get a much better idea, and there have been some very exciting leads in the world of research. On the one hand, are the things that that we can intervene on, and so one of my particular interests in my own research is how the local immune environment, those innate immune cells we were talking about before, we know that that the behavior of those cells can either promote the repair of myelin, or it can contribute to its ongoing injury. And we know that certain types of microglia and macrophages can actually promote the repair of myelin. And so, one of the strategies that's being taken is to find ways to modify the behavior of those cells to create an environment in which the precursors to myelin are triggered to reproduce and repair the damage that's been done. We also know that that the cells that form new myelin in some people just don't. They seem to be lacking in those signals that kind of get them to mature into myelinating cells. And so one of the strategies that's being done is to screen for drugs that help promote that sort of maturation into newly forming myelin. And then we also know that age seems to play a role in it. So younger patients tend to have more robust remyelination than older patients. And there may be a few reasons for that. But one of the potential reasons that's becoming clear in research is that as people age, number one, just the propensity of their myelin precursors to grow and mature is affected. It's something that we call senescence. And something similar happens with their immune cells, with those microglia and macrophages. And so we're identifying ways to kind of rejuvenate those cells so that they behave, more like, the situation in a young patient with MS than an older patient with MS. And so the three, big strategies are kind of impacting the local immune environment, finding drugs that promote the maturation and differentiation of these cells, and kind of rejuvenating them to what you might see in a younger person. And the last aspect of it, that I think is worth mentioning is that in order to get myelin repair, you have to have nerve cells there for them to myelinate. And so, just as important as remyelination is protecting those nerve cells and preventing them from dying, so that even later on in the disease course, you still have something to repair.
Some of your research is focused on neurodegeneration. Can you tell us what neurodegeneration is and what its role is in MS progression?
Yeah, so this is a nice segue into what we were just talking about. So neurodegeneration means the degeneration of nerve cells. So the nerve cells themselves, not just the myelin, but the nerve cells which are carrying the electrical signals in our brain and our spinal cord. In MS, those can get injured and eventually die off. We know that even early on in MS, people with MS tend to have more brain atrophy than people who don't. And that really accelerates in a lot of people with progressive MS. And once that's happened, then it is very hard to repair the damage because the substrate for what you're trying to repair is no longer there. And so we know that in progressive MS, the degree of neurodegeneration is really the primary predictor of the disability that people have. And so a major goal of research, both for me, but also broadly within the community of MS researchers, is how do we protect those nerve cells themselves, keep them healthy, so that we maintain function, we stop progression, and we maintain that substrate, that we can repair myelin with.
Now, I know your research also focuses on studying the role of diet and metabolism in inflammation. Can you explain how diet might affect someone's immune system?
Yeah, so to me, it's a topic I'm very passionate about. I find it fascinating. So, it turns out that over the last 20 years, researchers just in the lab, have found that when immune cells receive inflammatory signals and they become activated and angry and inflammatory, their metabolism changes in certain ways. They become much more reliant on sugar for their energy than other sources. And now, there's just been a very wide spectrum of changes in metabolism that we know occur when immune cells get activated. And it's been shown in animals and in cultured cells that if you can intervene on those metabolic changes to actually change the function of the immune system. And so from there, it's not a big extrapolation to think that we could do something similar with dietary interventions. The problem is that dietary studies are really difficult to do in people, and so at this point we really have very limited evidence in people of what specific diets are either good or bad for the immune system. So, I tell, all my, my patients that, I'm someone who's a believer that there's connections between diet and metabolism and the immune system. But it's important to understand that we don't yet know, which diets are good, which diets are bad. And so, when you look on the internet, you'll see all this information about this food is pro-inflammatory or this food is anti-inflammatory. And that goes well beyond, what we know at this point. But I'm certainly a believer that there are connections there.
As a result of the first 2 COVID-19 vaccines that received FDA approval, we've all become familiar with mRNA vaccines. Recently, researchers reported using a novel mRNA vaccine to treat mice with a mouse version of MS. Now, this study received a lot of attention because it uses the similar technology to the Pfizer-BioNTech and Moderna COVID-19 vaccines, but in this case, that technology is being applied to a different purpose. How does this mRNA vaccine work in mice?
Yeah, so I'm glad you brought that up. I think in the days after that study came out, I've never gotten so many messages about a paper that came out in a scientific journal from my patients as with this. And I think it's important to understand both the promise, but as well as the limitations and the gaps in that approach. So the idea is that in multiple sclerosis, people have these autoreactive immune cells floating around in their body that are designed to target myelin in their brain. And one approach has been to try to make those cells less reactive. So it turns out that we all, if you take any one of us who doesn't have MS, we all have cells in our body that are actually designed that are specifically against parts of myelin. It's just that in people without MS, they know that they need to be tolerant to that. That myelin is a friend and not a foe. And so the idea with this vaccine is to get the body to recognize that myelin is not the enemy. And the way it does this is by, just like with COVID, the mRNA encodes a protein that's derived from myelin, but it does it without any other inflammatory signals around. And sometimes when the immune system encounters something without any of those other danger signals, you can teach the immune system, that it's a friend and not a foe. And so that's the idea behind the approach. Aside from using vaccines, there are other groups that are using kind of tolerant immune cells to do the same thing. So basically injecting immune cells, into the body that kind of train the rest of the immune system on how to react to myelin. It's a very interesting approach, but the thing to remember is that the situation in mice is just so much simpler than it is in people. And particularly because the group that did that study in mice it's led by BioNTech, which produced the COVID vaccine, and the COVID vaccine was developed so quickly that I think people naturally think that that this sort of vaccine for MS is going to get to people in a similar time frame. And we have to remember that this animal model of MS is very simple. Mice are essentially immunized. They're given a protein derived from myelin in a way that creates an inflammatory response. And this vaccine was given to them to kind of calm that down. In humans, we don't know, what the specific proteins are that trigger the response. It's much more complicated. And so there's still a big gap between that study and when this is going to even hit clinical trials for people. It's also important to understand, that this would be an approach that, again, is really targeting inflammation. So it's meant to prevent future relapses. It is unlikely to repair damage that's already been done. And so it would be a treatment for relapsing MS. So it's intriguing. I really look forward to seeing how that sort of research develops in the future. But it's important to understand that it's far off from really having a role in the care of people with MS.
Well, we've covered a lot of ground today, which is great. I think shows the breadth of research that's being conducted to ultimately solve MS. Dr. Kornberg, what would you say are the top 3 takeaways that you'd like our audience to remember?
I think number one, just kind of putting everything into a framework. When it comes to disease-modifying therapies for MS, we have made tremendous progress in developing anti-inflammatory treatments that prevent new lesions and prevent new relapses. These have undoubtedly impacted the long-term outcomes for people with MS. But there's still room for better anti-inflammatory therapies. And so when we talk about stem cell transplant, or we talk about mRNA vaccines, there's still room for anti-inflammatory therapies that are either more effective or safer and more targeted. But what we don't yet have good options for, and what is the next frontier in MS research, is slowing or halting progression or promoting repair. That is kind of the next frontier in research. And so that's number one. Number two, I would say that even though progression and repair are more challenging problems, we have seen some great strides over the past several years in understanding, why progression happens, why repair fails, and understanding how we can measure these things in clinical trials, so that we can actually evaluate new therapies, and in developing candidate treatments that are at various stages of clinical development. So even though none of these therapies are quite ready for prime time, I am very optimistic, that within this next generation, we are going to see a number of these therapies succeed. And so I think it is a very exciting time in MS research. But I think we have to understand that some of these therapies we're going to be seeing on the order of years and not months. And then my final take-home point, which I really want to emphasize, is I always say to my patients that I can't understand what you're going through because I'm not going through it. But I have taken care of enough people with MS that I can certainly empathize with the frustration, with the despair that can come along with living with disability from MS, or having a loved one who has significant disability from MS. And because of that, I think there's a very understandable temptation to seek out experimental therapies that are being done outside of clinical studies or at unregulated facilities. And I just want to emphasize that it's very important, when we talk about these treatments that are in the pipeline, to pursue them within the confines of a controlled clinical study, for many reasons. Number one, they're called experimental therapies for a reason, and even though we think we have good reason to think they might be beneficial on people, have to understand that animals and people are much different, and there could be unexpected adverse effects that we just don't know about, and so you really need to pursue these within a clinical study. And if you're going to pursue something outside of a clinical study, you really need to make sure that you're going somewhere that is reputable, that is well regulated, and that can provide you some evidence of what they're doing, because number one, we want to keep our patients safe, and I think we've all seen some bad outcomes when people understandably are looking for things that are going to help them.
Well, I want to thank all of you who submitted your questions today, and I want to thank you, Dr. Kornberg, for being with us.
Absolutely, it's my pleasure.
I think that one of the best things that anyone can do for themselves, especially during these uncertain times, is to make sure that the information they're getting is credible and reliable. So we'd like to share some resources with you, and these are resources that you can count on. I want to remind you that the National MS Society's MS Navigator team is your best partner when it comes to connecting you to the very best information and resources on living with MS. You can reach an MS Navigator by phone, email, or through the Society's website by chat. And I'll also remind you that the MS Society brings people together through peer connection programs like self-help groups, the MS Friends Helpline and Paired program, and the National MS Society Facebook community. To learn more, please visit the Society's website at nationalmssociety.org/connections programs. For additional information on what's new in MS research, be sure to visit nationalmssociety.org/research. Every week on the Real Talk MS podcast, I continue the conversation that we start here. So I hope you'll take a few minutes and give Real Talk MS a listen. You'll find the Real Talk MS podcast at realtalkms.com, Apple Podcasts, Google Podcasts, Spotify, Amazon Music, wherever you find your favorite podcasts. Walk MS 2021 is coming, and this year Walk MS will meet you where you are to connect, celebrate, and make a difference like never before. For more information, be sure to visit walkms.org. You can connect with the National MS Society along with others affected by MS on Facebook, Instagram, Twitter, YouTube, and LinkedIn. I encourage you to like those posts, subscribe to those channels, and make sure that you're on the Society's mailing list so you'll continue to receive the latest information on MS and updates on upcoming programs like this one. I'd like to thank Dr. Michael Kornberg for joining us today. Please remember that a recording of this webinar will be available at the Society's website at nationalmssociety.org/MSExpert, as well as on Facebook and YouTube. And I hope you'll join us for next Friday's edition of Ask an MS Expert, when we're going to be talking with Dr. Adnan Soubey, Medical Director of the Multiple Sclerosis Program at the Memorial Neuroscience Institute, to learn how MS can impact your senses and to hear the best strategies and treatment options to manage those particular symptoms. And now I have a favor to ask each of you. Getting your feedback on today's webinar is important. So you'll see a survey pop up in a moment when we close out the webinar. If you're watching on Facebook Live, you'll see a link to that survey pinned to the comments section. And on YouTube, you'll find that link in the program description. Completing the survey makes a real difference. The information you provide helps us continuously improve, and it helps shape future programs. The survey takes just 1 minute, so I hope you'll take that minute to fill it out. On behalf of Dr. Michael Kornberg and the National MS Society, I want to thank you once again for joining us. Please stay safe and make healthy choices.